which I would like to briefly introduce you, I think we should try to have an hands-on on what are the approaches we would like to use for the running of the BigDFT code. I will briefly present what BigDFT code is, and then we will start by somehow using those tutorials. You will see they are based on the Google Colab. So you will have to run your tutorials on the cloud. There is nothing to install locally, which I believe can simplify the procedure. We hope that this procedure will go through smoothly. Then we will see if you have problems here and remotely. We are here to help. We will see the other 10 topics, which will be presented by Laura, Martina, William, Giuseppe, Sam, and the other people from BigDFT development group today. We will discuss about the so-called linear scaling part of BigDFT, which is something that enables calculation of very large systems. We will see what we could do with this part, why doing quantum mechanics for full quantum mechanics for full large systems. Here we are not in the QMMM idea, but we are in the idea of doing the entire system with only QM. Why this may be useful, that is something which we try to address in this section. And then we will see other advanced functionalities that can be performed with BigDFT code, like the inclusion of the implicit inclusion of the solvent around the molecule. You don't want to simulate the entire set of water molecules around a protein, for instance. You would like to have an effective treatment, and there are solutions for that. And we will see how the tools which were created in the context of this linear scaling BigDFT calculation can be employed to have a more efficient simulations of systems that are already split into fragments, or systems for which we want also to study the optical excitations under irradiation. This is a very dense program, we acknowledge that, so there is no need for you to catch all the details of all these tutorials. At the end of this section, we would like to have a brief walkthrough to the exercises to give you the idea of the take-home messages which each exercise will bring you. We are here to help, but of course you shouldn't worry if not all the points of the tutorials are completely clear. Okay, so I can move rapidly to the next, to the introductory part. We have been discussing about BigDFT, with BigDFT, always this name. What is BigDFT? It's an implementation of a density functional theory code under the formalisms of Dobeshi's wavelets. The wavelet is a basis set which was employed in signal processing. In the late 70s, it was used to find oil in the ground. So it's a basis set which is coming from applied mathematics, which presents interesting properties for DFT. We added these ideas in 2005, so it was now more than 15 years ago. At that time, most of the DFT codes you see mentioned here were already present and very well used. So there is a number of DFT codes that are available in the community. BigDFT is the only one which employs Dobeshi's wavelets, and each of the code is it's well suited for a set of applications and a set of, let's say, systems to be treated. The idea of using wavelets is related to the fact that they can refine the description only where it is needed. For instance, with wavelets, you can get an information like, sorry, I will skip this part, like here, where if you have a molecular system here, you may want to have a high resolution description of your system to express your wave functions, so the positions of the electrons. I do not know if you have the possibility to study quantum mechanics yet, but a wave function is a mathematical object that describes, it's related to the probability of finding an electron in a region of space. The probability is higher close to the nuclei, so this wave function is a function that oscillates a little more close to the nuclei. So it's important to have a higher resolution in the regions which are close to the nuclei, and it is not necessary to have it when you are a little far from the nuclei. In this context, the Dobeshi's wavelet formalism comes very handy because you can put your computational degrees of freedom only in the region where they are relevant. This saves memory, this saves computational time, and gives you the possibility to have a let's say, optimal description of the system, which you can improve. If you want to improve the description, you have two possibilities. You may decrease your grid spacing, because the wavelets are functions which are defined on the points of a grid. So if your grid is finer and finer, you will have more wavelets, so you will have a better description of your system. And you can also increase the radii, which define the blue and the yellow regions in this cartoon. If you have a very, very dense grid with a very large radii, you will tend to the complete solution. Of course, this will cost you computational power. You will have some exercises about these points in the first session of the tutorials. Now, that is essentially the main computational setup that the user of this kind of code has to trigger: the grid spacing and the extension of the region. When you do that, then you also have to decide, of course, the system you want to simulate. So you have to provide the atomic position. So you need to create the system. You want to simulate a water molecule, you have to tell to the code where the oxygen is, where the hydrogens are, in which position in the space, and also which are with what we call the boundary conditions. Is this water molecule alone in the universe? So what we call free boundary condition. There is only that, 3 atoms, and that's it. Is this water molecule periodically replicated? So then there will be the cell, the surface. We can also have a mixed boundary condition where we have periodicity in one direction and isolated the direction in the other. So essentially, you would get, I will come back to this slide after, you can get a description like this one here, where you can somehow mix systems that would have been otherwise more involved to simulate. If you have a code like a plane wave code, which is like VASP or Quantum Espresso, Abinit, those are code names you may have heard about. The boundary conditions in those codes are always periodic. So you have to decide which is the size of the cell which simulate, which is the basics of your simulation. This thing is not necessarily happening with BigDFT, where you can control the boundary condition of the system. Now, essentially, when you control the boundary condition, you have two sets of parameters, so the grid spacing and the extension of the grid. And you'll see that the less you increase, the more you increase the grid spacing, so the more you decrease you increase the density of the grid, the rapidly you go to a solution which have an absolute, let's say, precision. You could do the same with the sphere mount. Those plots would be similar to what we would do in the exercise. So essentially, this is the main idea which are beyond the formalism. We built this code, it was 2008, with this formalism in mind. It was possible to simulate different kind of systems. We started benchmarking the approach with respect to other DFT code which were present in the community. And we have been able to verify that indeed the features of the approach in terms of precision and reliability of the computation were very interesting. we have then included more and more functionalities to this code. Initially, it was started as a prototype, and then the code became a mature and, let's say, functionalities-rich code. Of course, we have limited manpower. We focused on the functionalities we thought was, let's say, specific to this formalism. And one of these functionalities, what we have been named linear scaling part of the code. This name is related to the fact that the overall computational time needed to perform a computation in those approaches is not scaling linearly with the number of atoms in general. So what happens in general is that if you want to double the size of your system, for those kind of code, you need to have 8 times the computational power. Those codes are what are usually called cubic scaling with respect to the number of atoms n. If you use another formalism, which could be built on top of codes like BigDFT, this computational scaling will grow linearly with the number of atoms. And that is potentially advantageous because you could go towards even larger systems than what could have been treated with the previously existing formalism. So we have been using BigDFT a lot for calculating small systems. With small, I mean a few dozens, 1,000, hundreds, few hundreds at most atoms, where the high precision of the description, thanks to the wavelet, is useful to explore the different local minima that a system may have by changing its configurations in the configuration space. You take the atomic position, you modify the atomic position, and you see whether you find better energy with respect to the previous one. This is what goes under the name of potential energy surface exploration. You can explore lots of local minima of a particular system. Here we have an example of a paper about boron 80 clusters, where we find that there are lots of disordered configuration in the system. So this is an approach where you use the high precision of the wavelet. But there is another approach where you use the potentialities of having lots of system and atoms that can be simulated. So essentially, thanks to the fact that the wavelets are a basis set which is localized in space, you can expand the information only where it is needed, but you can also design a set of degrees of freedom which are defined only around a small region. So essentially, you do what goes under the name of divide and conquer a system. This is another keyword that is very common in our community. You take a very large system, you split the system into portions, and each portion is simulated in, let's say, in the surrounding of the others. These portions are then based on what we call the support functions, which are instead of let's say, full wave function to the system, you split the wave function into many, let's say, sub-basis functions. So these are another set of basis functions, which is defined in wavelet, which are optimized in the context of the system, and that can be employed to express the full wave function at the end. So essentially, this is the main idea, which will be explained after in a dedicated set of tutorials, and Laura will have a talk about that, to somehow take profit of these function. And we will also see that it is not necessarily useful to use DFT in the very same way of potential energy surface exploration. You don't use DFT for 10,000 atoms the same way you would use it for 80 atoms. It's another way of using it because the number of configurations, the configuration space is exponentially growing. You don't want to deal with all the position of 10,000 atoms. You use DFT in another way when it comes to such large systems. Someone gives you the atomic position and you analyze the properties that this system will have under this quantum mechanical description. So we will discuss about these in the forthcoming hours. The point is now how the code is distributed. We have had to put this code into supercomputers. We will not have the time to discuss about how we have put containerization or how we have split the build system of the code. What it's important to know at this present is at this stage is that the BigDFT is a collection. This is of different computational libraries which are all linked together and can be installed in various ways.
There was a lot of information this morning, so don't worry if you didn't remember all of it. Obviously, the notebooks will be there, so the material is still available if you want to go back to it. But just to kind of summarize the main kind of take-home messages from this morning, you should remember that we have these three levels of atoms, fragments, and systems. And in this session and the session just after, you'll start to learn why we bother having this level of fragments. So why not just have atoms and systems, but we'll actually make use of them in this session, particularly in what William's going to talk to us about. We then have this calculator where you can actually run the DFT calculation. So this takes both the system and the input file. And as we've said in this tutorial, for none of the other calculations, you actually can be running them. We run them for you. But in principle, you could use the same notebooks to run the calculations, except for the kind of calculations that I'll be talking about in this talk, you would really need to use a supercomputer. So those are the kind of main things that you need to remember. So in this session, as Luigi said, we're going to talk about the big in BigDFT, so this linear scaling DFT. So the first question is, why do we need to go to such big systems? And big is always kind of relative as well. I've talked in conferences where I've said big systems, and then the next person's talking about kind of nanoscale with millions of atoms. So we don't mean millions of atoms. For some people, hundreds of atoms is already big if you're working with a very precise quantum chemistry approach. But in our case, big means kind of thousands or maybe tens of thousands. So I'll show some examples. And one of the reasons why we want to do big, so you saw yesterday about QMMM. Obviously, there are very many cases where this kind of approach works well. So why not just always use QMMM? Well, sometimes already you want the QM region to be big. There might be some reason why you need to include some combination of explicit and implicit solvent. There might be that you just have something happening in your system that already takes place over a large space. So you need to have a large QM region as well as the MM region. You also might want to test different ways of doing QMMM. So you might have done your QMMM code, but it's the first time you use that formalism, the first time looking at the system. Maybe it'd be useful to do a full QM calculation and compare the two to check that the QMM made sense. But sometimes also you just want to calculate things that you can only get from quantum mechanics, so things like excitations. Sometimes also you want to learn different types of things about the system. So we'll see particularly in William's talk, one of the things that we can do with BigDFT is to split up the system into fragments and do these kind of things automatically and analyze some of the interactions between fragments. And in that case, you kind of need to be working with a pure QM approach. So why can't we just use BigDFT as we've seen it so far? So just with our cubic scaling approach, where you just have the wavelets, everything kind of works pretty similarly apart from the wavelet basis to standard DFT codes. Well, we have a number of operations in BigDFT. It doesn't matter too much about the details, but the point is that eventually you hit some point where the cost scales with the cube at the number of atoms, as Luigi mentioned. So just to show an example for graphene, you can see that the time, I mean, actually, the time is maybe not too bad. If you've got enough supercomputing, you might be willing to wait an hour and a half for 1,000 atoms, depending on how many cores you've got. But actually, in many cases, what starts to become more of a problem is the memory. As you increase the number of atoms, eventually you get to some point where you just can't fit your calculation on a supercomputer. So we want a new approach that kind of bypasses this cubic scaling problem in both time and memory. So it's actually very well known and has been for quite a long time that the behavior of large systems is short-sighted or nearsighted. So this is the principle of nearsightedness. And if we want to kind of be a bit more precise, we can say that the density matrix decays exponentially in systems with a gap. And it also decays slightly different in metallic systems, but there is also a decay where you can add in a temperature. So we should have some way of being able to exploit this nearsightedness to look at large systems, which Luigi already hinted at in the first session. And the key idea is that we go from these cone sham orbitals, which are delocalized across our entire system. So here it's a small molecule, but you imagine, even in a protein, these will be delocalized across the whole system. And we work with something which is localized, which we call these support functions. So our cone-sham orbitals are basically some linear combination of these support functions. And then our task with linear scaling DFT is to find both the support functions and what the linear combination would be to construct these wave functions, but without having to explicitly construct the wave functions. So we have some extra steps. So we basically work around the density matrix, which if you would define the density matrix in this wave function approach, you have the wave functions, you have some occupations, which tell you, is there an electron or some fraction of electron in this orbital, or is it empty? Instead, we write this in terms of these support functions that I just mentioned, and then another quantity called the density kernel. So these are our two important ingredients for linear scaling BigDFT. If we work with this approach, we don't have to reference the orbitals at all. So we can avoid the diagonalization, which is one of the things that leads to cubic scaling. We also can skip this orthogonality requirement, which also can be expensive to impose, and we instead have some property called idempotency, which is basically depends on this kernel. And then the density you can reconstruct based on the density matrix. So don't worry too much about the details, but the important thing to remember is that we really care about the support functions and the density kernel. Those are our two key ingredients for linear scaling BigDFT. So first, the support functions. So these are atom-centered and they're strictly localized. So usually the radius will be somewhere between 6 and 8.4. And this is a user-defined parameter. So like with the grid spacing, where you can just make the grid spacing smaller and smaller to get to the complete solution, you can do a similar thing with the support functions, except you make this radius bigger and bigger and bigger. And eventually, if you would have an infinite radius, you should get the same result as cubic scaling BigDFT. Another important thing about the support functions is these are numerical functions expanded in wavelets. So they're basically some functions which live on a grid. So where before we have these two levels of wavelet grid, we still have the two levels, but then our functions also live in these spheres. So it's some numerical sphere centered around an atom, basically, and the function lives in here. So for those of you who know something about Gaussians, you could sort of think of these as contracted wavelets in the same way as you would think of contracted Gaussians. So it's a way of shrinking the full space of the wavelet grids down to some localized representation. And usually, although not always, we have a minimal basis. So we would just have one support function for hydrogens, four for carbon, nitrogen, oxygen, etc. And you already might, if you know something about Gaussians, raise the question, how on earth is this going to be accurate if we only have a minimal basis set? So there's an extra step that we need to do. So we start with an atomic orbital guess, so something which looks like your standard SPD orbitals. And then we, first of all, impose a confining potential, which we gradually decrease. And the point of this is just to make sure our support functions don't try and spread out. We want them to stay well localized within their spheres. You start to have problems if you have too much that builds up on the edge. And then the important point is that you minimize the total energy of the system with respect to the support functions. So what this kind of means is that we are optimizing the support functions in some way that will reflect the system itself. So they'll adapt to kind of their chemical environment. So just to show an example with this molecule, so we still have something that's S-like and P-like, but if you look at them, you can see that S orbitals are not spherical. If you look at S orbitals on different carbon atoms, you can see they all look slightly different, because they've relaxed to kind of adapt to the symmetry of their environment. And same with the p orbitals. And if you compare kind of the initial linear combination of atomic orbital guess to the optimized support functions, here we're just looking at a 1D projection, the changes are quite subtle, but it's important in what happens to the energy. So that the error in the energy will be quite large if you would just use a minimal LCAO basis. But once you do this optimization, the error becomes much smaller, and you end up with a minimal localized basis, which has an accuracy which is pretty similar to the wavelets. So that's the support functions, that's our first ingredient. And then our second ingredient was this density kernel. So we have a few different methods for getting the correct density kernel. So one is just doing diagonalization. So this is kind of useful for small systems and benchmarking, but the diagonalization has a cubic scaling cost, so that slightly defeats the object of working in a local system, although we have fewer degrees of freedom to do the diagonalization in. We also have what we call a direct minimization approach. So we still have the support functions, but then we go back to working with the cone-sham orbitals and work with those coefficients. So this is still, we're not completely getting rid of the coefficients, but it has an advantage that you can get some of the unoccupied states, which for certain applications we'll see later can be useful. And then our third method, which is the main, this is the linear scaling approach, that if you want to look at a system with 20,000 atoms, you're going to use this. This is the Fermi operator expansion method. The important point here is that it is linear scaling, and it can also work for metals, which are quite difficult to do with linear scaling approaches. You just have to impose this small finite temperature. And then we also impose some localization on the density kernel. We truncate it past some given radius. Usually this parameter is kind of set by default. You don't need to worry about it too much, but it is there and it is necessary if you want to be strictly linear scaling. So if we put these two ingredients together, this overview of our algorithm looks like this. We have our atomic orbital guess. We optimize the support functions a little bit. We optimize the kernel a little bit using one of the three methods. We repeat until both of these things are converged. So we have this outer loop, we have a support function loop, and we have a kernel loop. And all three of these things have some associated set of parameters. And then at the end, we get out the energy, we can get the forces, so you can do things like geometry optimizations and molecular dynamics if you want to. And then if you were using FOE, but you still want to, say, calculate the energy levels or do your density of states at the end, you can do just a single diagonalization, which, if it's only one diagonalization after a whole calculation, is not too much of a price to pay. And because we have localization everywhere in our system, we can use sparse matrix algebra, and that kind of allows us to have the linear scaling. One thing kind of important to note, so the sparsity would depend on a lot of factors. So it depends on obviously the size of the system. It depends on the dimensionality. So here I'm comparing a 1D nano ribbon to fully 2D graphene. And if you would look at what the density kernel looks like, you can see that the ribbon in 1D is much more sparse. We've got lots of more empty matrix elements than for the 2D system. And if we would look at for the periodic system, it will be even less sparse. And this is kind of useful to know, because if we plot both the time and memory, the cubic scaling versus linear scaling, you can see that in the ribbon, we only have to go up to a few hundred atoms before linear scaling is cheaper than cubic scaling. But in the 2D system, we already have to go up to 1,000 atoms before it's cheaper than the cubic scaling. And if it was a fully periodic system like diamond, for example, you might have to go up even larger. So just because we say linear scaling, this doesn't mean always cheaper. It means cheaper for some large enough system past the crossover point. And this crossover also depends on other parameters, the band gap, what you use for your localization window, etc. So one of the key parameters is the support function, these localization radii, how big are your spheres that the support functions live in. So you can choose different values for different atomic species. And as I mentioned, if you increase this enough, eventually you obtain the same solution as the cubic scaling code. But actually, in most cases, it's not really necessary to play too much with the details of this parameter. You can import what we call a profile, and you'll see how to do that in the tutorial, which basically imports kind of a set of parameters which are known to work well across a range of systems. So if you have a relatively well-behaved system, you might be able to just import the profile, and linear scaling BigDFT will work more or less out of the box. So because the linear scaling uses the same kind of infrastructure as the cubic scaling code, but with this addition of a profile, we inherit many of the same features. So we still have the range of boundary conditions. So these are all outputs from linear scaling calculations, 1D, 2D, 3D, and in this case also a metal. But we don't have all of the same functionalities of cubic scaling code. So just in case, for those of you who know about DFT, we can't do hybrid functionals with the linear scaling code. That's only in cubic scaling. We can't do time-dependent DFT with the linear scaling code. That's also only cubic scaling. So depending on the functionality, it might not necessarily be available in linear scaling DFT. But what we can do is very large systems. And so here, just to show that, or I've shown you the linear scaling behavior for a smaller system, but it's linear scaling in this case for DFT, and this plot's already for DNA, sorry, it's already a few years old. You can see we can go up to the tens of thousands of atoms regime. And just recently, we managed to converge the COVID spike. So this was combining linear scaling DFT with the implicit solvent that Giuseppe is going to tell us about later. And so here we can do 50,000 atoms in about 6 hours and 128 nodes. So really, you can do purely QM calculations of very large systems without too exorbitant supercomputer resources required. And once you can do this kind of system, so you can see there's some something going on with the coloring here. So this is based on a particular type of fragment-based analysis we can do, which William is going to tell you about now.
Just a few words. We have seen what happens when it comes to large systems. The formalism is not exactly the same. You know, there are an extra layer, which is the support function basis, which turns out to be handy for two reasons. First, because it gives you the access to linear scaling algorithms. And second, because it embodies the actual property of the electronic structure of the system. Those support functions are optimized around the chemical surroundings of each atoms, which means that they encode something that is proper to a given system. They are not decided before. So we got these are not things which are really unique to BigDFT. There are other codes like OneTap, which is developed here in the UK, that employ the same concept. Here we are going a little further, and we have been trying to ask ourselves whether the presence of those optimized degrees of freedom is actually telling us something more to the system. And we are going into another approach. What we are going to illustrate now is something that is more specific to our experience, is related to the BigDFT consortium, even though the concept can be employed with other codes. This idea is related to our own questions. We have been trying to ask ourselves what would be the reason to perform such kind of calculations. Why one should tackle the entire spike of SARS-CoV-2 with a full QM method, and what can be extracted out of it. So what now William, which is, as we speak, is in Kobe, in Japan, is going to present us, is associated to such a kind of questions we have been trying to ask ourselves. And there will be also a dedicated tutorial associated to that, which will be performed after the linear scaling part, either after lunch or before lunch. It will depend on what we are going to do in the next minutes. Please, William, you can continue. Go ahead.
Okay, so before I start, I just wanted to mention that some of us who are participating remotely, we can see the conference room, but we can't see any of your faces. There's these big computer screens. I see those, and I hope behind those there's some smiles perhaps, but so we're moving now away from the basics of the code to some of the more advanced features and application-oriented features. And this one is called complexity reduction. So, oh, there we go. All right, so you should know historically that for many people who have done DFT calculations, the impression is that you can only treat small systems. And this is the impression they had because for many years this was the case. You really could just treat systems made up of tens or hundreds of atoms with a quantum mechanical method. And so I think about one of my collaborators. He said to me, you know, I want to study lignin. And what he did is he built this little model of lignin right here. He put all of his chemical knowledge into this little model and ran a bunch of calculations on this little system right here. And that's all he could do. But he was able to learn a lot about the big lignin system by studying this little thing. Nonetheless, for those of us who enter the field later, when we see that now with modern codes like BigDFT, you can treat big systems, our temptation is to say, no, I don't want to know anything about chemistry. I don't want to build this little model. I'm just going to treat the exact thing that I want to study. So this huge system, it could be a protein, it could be some sort of material, but I don't want to cut down the scale. So you might be tempted to do that too after seeing Laura's talk. You might think, well, if the code can really treat the spike protein, I don't want to learn anything about how to cut down a system. I just want to go straight to that. But I think we should give you some caveats. The first thing is if you want to treat a big system like this, it's going to take you a lot of work just to even set up the system you're studying, because it's big. And that's why we introduced all these things about systems and fragments. It's why we're using Python for so much, is that if you want to set up a big system, you have to be prepared to take some time putting together the pieces. And we hope that the code we provide is of some use to you in that. The other thing is, if you maybe have some experience treating big systems from using force fields, and you should know there are some key differences between force fields and DFT. The first one is that all the atoms have to be there. A lot of force fields, you can do a type of coarse graining, or you don't really care about hydrogen so much, but DFT cares about all the atoms, and it also cares about all the electrons. So you better tell the system, the code, how many electrons are there. Right now, when you run it in these notebooks, we haven't done that, and that's because we're assuming the system is neutral. But as you might know, many big proteins have a net charge on them. They have more electrons than would be there by default, and you need to pay attention to that. And the other thing is that if you want to treat a big system like that, it has to be a good geometry to start with, because as Luigi mentioned, if you have a strange configuration of these atoms, the electrons will really struggle to figure out where to go, and then you're wasting time just waiting for the calculation to run. One other caveat is that the calculations take a long time. Laura showed you the calculation on the spike and said it takes 6 hours, which for many people who do DFT might think is impressive, but you should remember that that's just for one calculation. So you're not going to do things like molecular dynamics. You're not going to run 1 million snapshots because each one would take 6 hours on a big machine. So just to summarize these things, please do not think about DFT as just a super force field. You can't just swap it in for whatever your force field workflow was, or your QMMM workflow. Instead, you should be thinking, can DFT serve a different type of niche? And that's what our complexity reduction framework is aimed at. So as we've mentioned many times, the thing that's different about DFT than force fields is that we explicitly include the electrons in the calculation. And so if you actually run the code, you can get out some picture like this, where you actually see where these electrons live. You can plot it out and take a picture of it. So this is something you couldn't do with a force field, and you should start thinking, okay, this is an extra piece of information I get from a DFT calculation. What can I do with this extra information, these electrons, to try to understand the system better from a different point of view than if you were using the standard techniques you might be used to. So one way to look at these electrons is through this density matrix that Laura introduced in the last talk. This is a matrix that gets spit out of any DFT calculation that you run with BigDFT. It's an n by n matrix where n is the number of those support functions that Laura discussed. And it will look something like this. Each of these support functions, as Laura said, are associated with a given atom. Some atoms have 1, some atoms have 4, etc. So you should actually be able to take this matrix and divide it up into, different rows are associated with different atoms. So in the case of a protein, you could even say this block of the matrix is associated with this amino acid, and this block is associated with this one, and this block, these off-diagonal blocks, are associated with pairs of amino acids. And since Laura mentioned, if you look at it, it actually has this sparse structure where if you have amino acids that are far away from each other, for example, 1 and 5 in this case, because it's looping back around, you'll look and you'll see that there's not so many elements there that have a large value. And so we were looking at this matrix for a while, and then we thought, maybe this can tell us something about the system that we didn't already know. It actually is saying to us that you should be able to break down a system and use this sparsity as a guide to tell you what are the building blocks of the system and how much they interact. So one of the properties of this matrix is that it's idempotent, which means if you multiply the matrix by itself, it should come out the same. So we thought, okay, let's try to exploit this property for building an analysis technique. So if you take this matrix, which I showed you right here, and instead of splitting it up into amino acids, let's just split it up into two fragments. So you have A and B. Now, if you took these blocks right here and you said they're associated with part A, and this one is associated with part B, BB, well, it would not, these blocks would no longer have that property, that idempotency property on their own. And that's because if you follow the block matrix multiplication, you multiply AA by itself, you would have neglected these off-diagonal terms. And so we were thinking about this and we said, if these off-diagonal terms are very small, it would almost sort of imply that this block, these A block, doesn't have much interaction with B. And in fact, we can measure the amount of how much is there by looking at that idempotency, by doing that multiplication, and you do a trace operation, some linear algebra, and out comes two different measures. The first one is called the purity value, which describes how good a fragment is based on some measure of these blocks. And the second one is the fragment bond order, which defines the interaction strength between two blocks. And what we can do is we can use these measures in order to break a system down into its fragments that make it up. They could be amino acids, or they could be something more coarse-grained. Maybe you don't want to think about a protein in terms of the amino acids, or maybe it's some system you don't know how to break down at all. It's just this big complicated mess. Well, you can use these measures to do that. And you can also use it to generate these kind of graph views of systems, which show you which parts, once you define them, are interacting with each other. Okay, so I'm just going to tease you a bit before we go back to the tutorials and show you one of the things that we recently did with this framework. In this case, we are interested in studying the spike protein of SARS-CoV-2. And one of the things that's interesting about SARS-CoV-2 and very, I say interesting, but very troubling about it, is that it can evolve. And what that means in practice from our atom point of view is that there are amino acids, there are atoms in this system that get swapped out for another set. So in the case, one very common mutation that exists in the beta and the gamma strands is this mutation at point 484, which switches from glutamic acid to lysine. And so we thought, all right, we can calculate this system with BigDFT. And in fact, we can calculate that spike bound to human ACE2, so where it's trying to get into your cells. And we could actually look at what happens if you modify those atomic positions so that you switch from glutamic acid to lysine. So you actually enforce this mutation. And then we post-process it with that framework I vaguely introduced in the past slide, that complexity reduction one. And what comes out, and I hope the Zoom is working here. Can you see the Zoom as well?
Yeah.
Well, yes, okay, thank you. So what comes out is these graphs that I promised, where you have, in this case, you can look at the wild type, or you could get the graph of the mutated version. And the interesting thing we saw when we looked at it is that this E switches to K, and actually it goes from red to blue, which says it was pushing away before, but this framework said actually now it's pushing it together. And these are the kind of things that you can identify with this framework, and we'll show you how to do a bit of it in the tutorial that's upcoming. Okay, thank you.
Yes. Can you see my slide?
Yes.
Okay. So good afternoon. I am Giuseppe Fisicaro from CNRMM of Catania. I will show you the theory behind this implicit solvation approach that we implemented in BigDFT. It's named soft sphere. There is a lot of math, but I will not go on the very details, but just I will give you a general picture essentially. So this is the overview, some motivation, then some details on the electrostatic solvers and on the soft sphere model, and then some application to particular molecular adsorption at some solid-liquid interface. About the motivation, we know that a lot of processes take place in a solvent environment, both neutral and an electrolyte. This happens for the synthesis or growth of material, solar energy harvesting, electrocatalytic water splitting, and so on and so forth. So to support this kind of study, it is important to include the environment in our simulation, especially in ab initio atomistic simulation. This can be done, just let me, I'd like to take the pointer. Okay, let's continue. Can you see my hand in the screen?
Yes.
Okay, so here you can see we have the explicit inclusion of the solvent. So this green sphere, it's our solute, and then we have all the molecule of the solvent. So in principle, this explicit inclusion of the environment should be the best way, no? But on the other side, we have several problems. One of the main problems is that the size of the system easily became very big, very large, impossible to forward computation from a computational point of view. Think about of this spike variation that Luigi showed you with something like 50,000 atoms. If you want to fully solvate this molecule, I don't know how many atoms you will reach at the end. So possibility is to describe this interaction of the solvent with a mean field. Mean field is like a continuous function, homogeneous and isotropic in the three-dimensional space. And you can see here, essentially, this is just a schematic, but we replace the explicit solvent here with this mean field, which is a continuous function. It's described by a permittivity. And now the degree of freedom of the system is exactly the same like in vacuum. So we don't have any explicit solvent molecule in our system. But however, in some way, we are able to include this interaction between the solute and the solvent. So we can simulate a really large system, include the effect of the environment or a wet environment. But we also can do extensive simulation like structure prediction or molecular dynamics in this way, since we are really reducing the degree of freedom of our system. So to build this kind of implicit solvation approach, what we need? We need a solver for a receipt for a cavity, no? Essentially, this function, this mean field that describes the environment, depend on the system, on the coordinates of the atoms, and this function takes the value of 1 where we solve the quantum mechanical problem, like we do in vacuum, and here we just take the value of the bulk and the electric constant of the solvent. So we need a receipt to build this three-dimensional cavity for our system. Then we need a solver for a generalized Poisson equation. You see here we have the permittivity, which vary in the space. So we need a solver for this equation, and then we need a model for the non-electrostatic terms to the energy. In principle, this talk I am supposed to give details in all these three components. I will go fast, but stop me if you want more details. So when we try to solve a problem in vacuum, this is the total energy of our system. It's composed on the kinetic energy, the interaction with an external potential, then we also the electrostatic energy here. So essentially what we do, we solve the standard Poisson equation, which is this one. In this case, we assume we solve the problem in vacuum. But if we have a wet environment described by the implicit approach, if the solvent is neutral, then we need to solve the generalized Poisson equation. Otherwise, the Poisson-Boltzmann one. The Poisson-Boltzmann essentially is identical to the generalized Poisson equation. We just add here on the right This is the electronic charge density. We just add the charge of the ions in the electrolyte here. But again, we need to solve a generalized Poisson equation. To solve this equation, we tried different solutions. This is a self-consistent approach, and this is the algorithm. Essentially, if we apply some math, we rewrite this equation, we recover this vacuum-like problem, and we are able to solve this equation in a self-consistent manner between this term, which depends on the potential, and the potential here. But we developed, since it exists an action integral, Euler-Lagrange equation is exactly the generalized Poisson equation. We can apply some minimization procedure, and we apply the preconditional steep descent or a preconditional conjugate gradient. So at this stage, I will go a bit faster. Here we tested our algorithm with analytic function. Essentially, this is the a Gaussian for the potential. Then we used an error function for the cavity, and then the resulting density applying the generalized Poisson operator. Then we need to describe the operator, and then we just compare, giving as input the charge density, which is analytic. We compare the analytic potential that we know already is the solution with the numerical solution. Here are the results. You can see the solver is able to solve the equation in some 10 iterations, reaching an accuracy of 10 to the minus 10. And this for free surface and a periodic boundary condition. Then here the solver for the Poisson-Boltzmann. We have two types of Poisson-Boltzmann, the traditional one and the modified Poisson-Boltzmann, which take into account the steric force and the finite size effects. And we applied the preconditional conjugate gradient method for the linear version of the Boltzmann. In principle, this should be enough for, let's say, for example, for a protein plunged in a solvent. We don't need to solve the fully Poisson-Boltzmann or the modified Poisson-Boltzmann, because the electric energy is lower than the thermal energy in this case. And this is a solver for the modified Poisson-Boltzmann. Again, we have a good accuracy. We solve the problem in some 5 iterations. Then we need a receipt for the cavity. Our cavity essentially is done by atomic-centered interlocking sphere, as you can see here. And the spheres are analytic, continuous, and differentiable. This means that their product, as you can see here, it's also continuous differentiable. And the cavity takes the value of 1 here inside the atomistic system, and outside it's just the dielectric constant of the solvent. So here, essentially, then we need a model for the non-electrostatic energy. Why? Because if we include only the cavity, we calculated the total Cauchy energy, and then we subtract the total energy in vacuum, this represents the electrostatic solvation energy. But in order to compare with the experimental solvation energy, we need to add the cost of energy to build the cavity, the repulsion energy, which in some way mimic the exclusion Pauli principle, and the dissolution energy, which account for the van der Waals interaction between the solvent and the solute. Now, we demonstrate in our paper that all this non-electrostatic contribution can be modeled as a linear function of the quantum surface and the quantum volume of the cavity. The volume essentially it's what is here inside. The surface, it's the surface of the cavity. It's trivial. And with this, the model it's completed. We just can calibrate the model against a database of free energy, experimental free energy observation of 274 organic molecule in water. And this is the accuracy of our model. This is the mean absolute error in kilocalorie per mole. It's on the same order of other approach. Then let me show you some application. Here we applied this implicit solvation study the solid liquid interface of calcium fluoride with water. Essentially, we found in vacuum that the 111 termination of calcium fluoride is lower in energy than the 100, and we have a plethora reconstruction for the 100 in vacuum. When we included the solvent described by the soft sphere implicit solvation model, we found that the wet environments promoted the growth towards the 100 surface instead of the 111, and this is confirmed from an experimental point of view. You can see the square shape of this nanoparticle by scanning electron microscopy. Then we studied the molecular doping of silicon. Essentially, people try to dope the silicon with putting the silicon in contact with a solvent, mesitylene in this case. So in this case, we parameterize the soft sphere model with another solvent that is not water, it's mesitylene. And we studied the absorption process of this complex molecule on top of the surface and the full path from the absorption to the breaking of the molecule and the transfer of the phosphorus atoms to the substrate. Here we include in this way, thanks to the soft sphere model, the effect of the environment. Last slide, here we try to study the interface between anatase titanium dioxide 101 surface and water and ethanol. And again, we assess the methodology because we compare the several quantity calculated both in implicit with implicit approach and the explicit molecular dynamics. And then we studied the molecular dissociative absorption at the perfect and the defective surface. The paper, of course, you have all the results, and these slides will be available in the school project of BigDFT if you want to follow more the details. So in conclusion, we build a solver for the generalized Poisson equation, the Poisson-Boltzmann. I developed an implicit solvation approach. It's fully integrated inside the BigDFT suite. We parameterized the model and we applied it to various molecular absorption process for different solid liquid interface. So thank you for the attention. This I finished my talk.
Thanks to Giuseppe. So the idea of this talk is to show that even though you can treat large systems, you always have to consider the environment in which this system is embedded. Sometimes it's important, sometimes not, sometimes it's fundamental. For instance, just anecdotic data, you have seen by Laura a picture of the calculation of the full spike protein of BigDFT. I spent many weeks in trying to make this calculation working, and I even eventually realized that the calculation was not able to converge, remember this plot, only because I forget the implicit solvent in the result. It was very funny to see that by including the implicit solvent in the calculation, the code itself was able to converge. And this is not unreasonable because these positions, the very spike position, was extracted from a molecular dynamics calculation in Solvent. And this answers the questions that Giuseppe asked during his talk. How many molecules would we have, how many atoms would we have had if the solvent would have been considered explicitly? I know this answer, it's 500,000. So even though the spike is 52,000 atoms and it's feasible, one thing is 52,000 atoms, one thing is 500,000. In a linear scaling code, it's a 10-time difference. So it changes a lot your results, especially when it is feasible and indeed the code itself is telling you that this is an approximation that is not only enough, but it's only mandatory for your calculation to get an interesting answer. So you have also an idea that this is not coming out of the blue. Giuseppe has made a very careful set of parameterization. You just you have to select a cavity, which is an effective treatment. You have to decide these parameters, which alter potentially the results of the physics that you can obtain. So in the forthcoming notebook, you have some examples.
You might think that you've heard everything there is to know about fragments in BigDFT. That's not true. I'm here to tell you how we can use fragments in another way. So this talk we're splitting between me and Martina's going to talk also about, once I tell you about this new way of using fragments, how we can also use that to calculate excited states in large systems. So fragments. So we've talked a bit about how the linear scaling calculation takes longer than cubic scaling for large systems. And one of the reasons why there's kind of a higher overhead than you maybe might naively hope is because of this support function optimization, which takes, depending on your system, a not negligible amount of time to do the support function optimization. But one thing I also told you is that the support functions adapt to their chemical environment. So you might ask the question, what happens if you have similar chemical environments? So if we look at this water droplet, for example, these are some optimized support functions from quite an old calculation. But if you look at them, at least as far as kind of squinting goes, they look pretty similar between the different molecules. And similarly, if we have here, this is a fully periodic silicon carbon nanotube. If you again look at the support functions down the tube on the same atom, they look very, very similar. So the question we asked ourselves some years back now is, can we somehow make use of this to reuse the support functions in some way and reduce the computational cost? So in this talk, we're talking about fragments as a way of making things cheaper, not necessarily fragments as a way of analyzing the system. So what we would like to do is something that looks like this schematic here. So we would take a template fragment. I always use the water droplet as an example. It's actually not a great example because water in gas phase, you would be better to actually do this in some implicit solvent. But let's imagine for now that this works as just a toy model. If you would take your template fragment, in this case a separable molecule, but you'll see later that it doesn't have to be the case. We do a full normal linear scaling calculation. We optimize the support functions with both of these loops. This is cheap, it's a small calculation. Then we would take our larger system, which is made up of some kind of repeating fragments. So in this case, several other water molecules. We want to somehow take these support functions from the template calculation and use them as a fixed basis in the larger system. And if we could do that, we'd be able to get rid of this support function optimization loop and bring down the computational cost by potentially quite a lot. So there's one kind of key ingredient that you need for this. So because these are support functions which live on a grid, it's not a simple matter of just rotating them. First of all, we need to figure out how to kind of pick up and move these support functions in a way that doesn't require us to do any thinking. And then we also need to actually do this rotating on the grid. So it turns out this is a very well-known problem. We have some cost function, J, which is often known as Waber's cost function. And this basically gives us some rotation matrix between a template set of coordinates and a system set of coordinates. And if you minimize this cost function, which is quick and easy to do, there are ways, known ways of doing this, it gives you the optimal rotation matrix. And so this means that we can just define in our input file, so we have our template system, we have our full system, and BigDFT uses this to automatically detect the rotations. Once we have the rotations, we then have a wavelet interpolation, which we use in what we call a reformatting procedure, which takes from the template to these various different roto translations. So because this takes place on a grid, you do have an egg box effect. So this means that if you would shift the molecule slightly with respect to the grid, this changes slightly the result. But if your grid spacing is small enough, this is not too big of a problem. But just one other interesting thing to note about this cost function. So if you have a perfectly rigid transformation, so your water molecules are not allowed to vibrate or kind of do anything interesting, and you're just rigidly rotating, when you minimize it, you'll get a value of 0. If there's some kind of noise, they've been fluctuating in their droplet, then the cost function will be non-zero. So the larger the cost function, the bigger the distortions. The bigger the distortions, the less appropriate this approach is. But this cost function J, which is based purely on the geometry, already gives you some idea of whether or not this could be a good idea. So this is suitable in two kind of limits. So in the first case, for relatively weakly interacting fragments. So here I've just shown the energy curve for a water dimer. So we have the cubic scaling result, the linear scaling result, which you can see agree very well. If we then have the fragment approach with a minimal basis, so this is the number of support functions on the hydrogen and the oxygen atoms, you can see that at long distances, great, works perfectly. At short distances, you start to get a bigger error. So this is because of what we call basis set superposition error, but essentially if we're generating our support functions for isolated molecules, it's not reasonable to expect that they're going to be very good if your water molecules are right next to each other. You can mitigate this slightly by having more support functions, or you can just kind of work in the regime where your fragments are relatively well separated. And then, as I mentioned, we also want to work with fragments which are not too distorted, so we can use this cost function J to tell us if that's true. If you're kind of close to the regime where this approach might work, but it's not quite good enough for out of the box, you can also use the fragment approach to generate an input guess for linear scaling BigDFT, which is one of the things we explain in the tutorials. And that just means you would do a fragment calculation first, but then you wouldn't fix the support functions, you would optimize them a bit further. So that can still save you some cost, but not quite as much as if you would keep them fixed and not change them at all. So the question is then, what happens if we want to look at an extended system? So a case where we don't have nicely separated molecules. In fact, in the most extreme case here, showing this nanotube again, how can we divide something like this into fragments? So we still have some repeat units and support function similarity that we want to make use of. And actually, we can even go beyond our kind of squinting at the support functions and quantify this similarity using a measure that we call the on-site overlap matrix. And this is basically if I pick up a support function, put it on top of another one, what's the actual overlap? So an overlap of 1 would be identical, 0 would mean they have nothing in common. And if you plot the on-site overlap along this nanotube, you can see that they are indeed all very, very similar. So in this case, what we decided to do is to take a slice. So we imagine a slice of the nanotube is a fragment, which is going to be replicated along the tube. But we can't just generate the support functions for an isolated slice because that wouldn't be very good representation. Instead, we would start with a small nanotube, in this case, or in general, a small representative system, and we generate our support functions embedded in this small system. And then otherwise, we follow the same procedure, we replicate them, and for our larger system, we can do fixed support functions. So the advantage of this is that we can cut covalent bonds between fragments, so we don't have to have a separable fragment. You don't need this to be pure in the sense that we saw from William earlier. But on the other hand, you have to have a bigger template calculation. So you can't just have your isolated fragment. You need to have kind of the smallest possible system that kind of has a representative environment. Of course, we also might have more complicated cases. So already, just if we would have a finite nanotube, which has some kind of change in shape as it reconstructs at the ends of the nanotube. And in this case, we might need different types of fragments. So if you look very, very closely, you can kind of see the support functions on the atoms at the end of the tube are slightly different to those in the middle. So if we would again plot this on-site overlap, we can see this kind of more quantifiably, that all the support functions that are kind of blue and purple are very similar, whereas the red ones are quite different. So here we kind of set up different types of fragments. So in our kind of simplest case, we would say we have the red ones, which are edge type fragments, the yellow ones, which are bulk type fragments, and then we replicate this for a larger system. This might not be accurate enough, depending on how much the edges kind of have a long distance effect. And in that case, we can define as many fragment types as we need. Of course, in this case, it means we need to do a larger template calculation, so things get a little bit more expensive. But in principle, you can generalize this type of approach to any kind of system you like. The fragments can be slices of a nanotube, they could be just individual atoms, they could be whatever you choose to define. It's really slow to load the next page. Yeah, so just to kind of summarize, we have these two different approaches to fragments, so that the molecular kind of fragments side, where we want to have weak interactions between the fragments, and embedded fragments where we're allowed to have stronger interactions between the fragments. So it could be the nanotubes, it could be also electric graphene, or it could be something in a biological context where you had, say, the amino acids strongly interacting with each other, and you wanted to use those as fragments. On the plus side for molecular fragments, we can do a smaller template calculation, either gas phase or something which we haven't tried, but which would make sense to do, is to use implicit solvent to generate the template. Whereas in the embedded fragments, you need a larger template calculation that has more than just the fragment, but enough of an environment to be representative. And we also have this cost function and this on-site overlap matrix to kind of tell us something about whether or not the fragment approach is appropriate to use. So, well, how does it actually work in terms of cost? So if we kind of summarize the methods available in BigDFT, looking at both the length scale and the level of approximation, we have, of course, at the bottom, the smallest systems we can do, you know, up to maybe about 1,000, depending on the system, with our cubic scaling. Once we impose localization, we got to linear scaling, which can take us to much larger. This was a pessimistic view, I guess, figure I made a few years ago. And then at the next level, we have fragments where we're introducing this fixed basis, and then after that, you know, we have implicit solvent. And if you would look at the cost, so this is for a particular type of OLED molecule going up to 5,000 atoms. You can see here the crossover between cubic and linear was about 1,000 atoms. But once we have the fragment approach, we're still linear scaling, but a much smaller prefactor. So I think for the largest system, it's a speed up of about a factor of 7. So if you do have a system where this kind of approach works well, you can get quite substantial speed ups. If you were doing something like the fragment guess, so it didn't quite work with fragments itself, you might kind of get maybe a factor of 2, depending on the particular details of the system. So what else can we do once we've kind of used this fragment definition? So the thing that we initially implemented at the same time with fragments was constrained DFT. And the idea of constrained DFT is that we want to find the lowest energy state, but satisfying some kind of constraint. And in our case, we're interested in a charge constraint on the density. So this is useful for treating charge transfer excitations, where you know that, say, one electron is going to move from one constituent of your system to another constituent. And the nice thing about constrained DFT is its computational cost is similar to the ground state. So this is much cheaper than doing something like time-dependent DFT, for example, which is particularly appealing for big systems. So the way it works is you have your Kernel energy functional, and then you add this extra term, which has a Lagrange multiplier. This K is just our usual density kernel. And then this W is a weight matrix, which tells you where you want this charge to go. So in our case, we're basically going to say we want some charge to be associated with a fragment. And then N is the charge that you actually want to transfer between fragments or onto a particular fragment. And so the task is to find the appropriate value for the Lagrange multiplier that matches this charge. In the support function approach, we define a weight matrix like this. So this is based on Loudon, for those of you who know Loudon approach. And this enables us to do what I just said. So associate the weight function with a given fragment. So that the accuracy of doing this kind of constrained DFT with fragments, of course, depends on being in a good regime where the fragment approach works. So if your fragments are interacting too strongly or too distorted, this is not going to work so well. And then it also depends on a second factor. So here, I kind of said so it depends on the suitability of the support functions for charge fragments. So if you've done your template calculation for a neutral calculation and then you want to add a charge to a fragment, you need the basis to be able to handle that as well. And usually what that involves is having a basis that also represents the virtual state. So if you remember in the talk earlier, I said this doesn't happen by default in linear scaling BigDFT, so you need to kind of do the extra calculation steps to make this work. So just to show the original application that we looked at for this was looking at an organic LED system. So we had this host CBP molecule and then doped it with this iridium PPY3, which is a common kind of guest emitter. And we wanted to look at various charge transport parameters, so that the on-site energies and the transfer integrals. The details are maybe not so important. But one of the nice things is that we calculated these on-site energies with constrained DFT. It's basically just the difference in energy between when you constrain a charge on a fragment embedded in its environment. And you can understand things like the effect of the disorder and also the effect of the environment or these parameters. So this works very well in kind of large systems. But I mentioned that constrained DFT here, this is kind of its standard implementation. It's good for charge transfer excitations. But in some cases, we might also want to look at locally excited states, and that's something that we've been working on a lot. So that kind of motivated us to look at a new version of constrained, or develop a new version of constrained DFT, which we call transition-based constrained DFT. And I'm going to hand over to Martina to explain how that works.
Okay, hello everybody. So just to give a little bit of context, we are now moving on to trying to model a mechanism that is no longer constrained to be on the ground state, but we are maybe interested in model excited states, which are mechanisms in which maybe an electron or a charge gets excited to higher energy level. And modeling this kind of mechanisms with DFT is a challenging thing. But there are a number of variations of DFT that have been developed in history to model excited states. One of these is the one that Laura has mentioned earlier, which is the standard constrained DFT. But what has been seen when trying to model excited states is that one of the main problems is that not all of the variations of DFT performs at the same way. And actually, depending on the kind of excitation that you want to model, you will have to choose a particular variation of DFT. That's because sometimes they are based on particular approximations or because of the way the formalism has been developed. So for instance, the constrained DFT that Laura has presented 2 slides ago is a cheap DFT-based method, but is very good for modeling excitations that are called charge transfer, which are excitations in which we assume that the charge is completely being transferred in between either 2 orbitals or 2 places, 2 different sections or fragments of the molecule. When it comes to a different kind of excitation that we call local, for instance, there's another variation of DFT that is very well known, which is called time-dependent density functional theory. This is very much used to model local excitations for molecules and larger systems as well. The problem of this method is that it's very expensive. Well, it is expensive, and so you can't really push it to very, very large systems in standard implementations. And it has issues with modeling charge transfer states with standard functionals. So for this reason, other methods have also historically been used for excited states. One of these is delta SCF. Delta SCF is probably the simplest DFT-based method that one could use to do excited states. It's a cheap method. It's basically the same computational cost as a CDFT. It's basically the cost of 2 single point calculations. It has a number of problems as well related with local minimum and spin contamination. Point is there's no one single choice to do excited states. So we decided to think about, to take the positives of the frameworks of the standard constrained DFT to present a new variation of constrained DFT that is called transition-based constrained DFT, with the idea to develop a method that would be flexible enough to treat charge transfer states as well as local excitations in large systems. So the main, very simple idea behind the transition-based constrained DFT is that instead of having a spatial constraint, so instead of having a constraint that forces the charge to be localized on a specific part of the molecule or of the system, we constrain a specific transition. So we constrain a specific, for instance, homolumo excitation. This, I don't want to go in the details too much of this implementation, but the good thing is that itself includes self-consistency. So it means that it gives us a description that is beyond linear response. And after we have benchmarked this on a number of cases that I'm going to show you, it showed to be flexible with treating local excitation as well as charge transfer. And it's being developed already within the fragment approach framework as well as linear scaling formalism, which means that it can naturally go towards the application on large systems. Yes. Okay, so when you think about an excitation, what I was saying, in the simplest possible way, we're thinking about, for instance, an electron that gets excited from an occupied orbital, often the highest occupied molecular orbital, HOMO, to the first empty orbital, which is the lowest unoccupied molecular orbital. So the simplest transition you can think of is a HOMO-LUMO transition, which is the first thing we try to simulate. In fact, in the simplest implementation, we have tested the method towards assuming that there is a single HOMO-LUMO transition. So we only transfer one electron. the way we do the constraint is to introduce Lagrange multiplier into the density minimization. So we constrain the density to have a specific transition. And the good thing about the method is that we don't need to spend time trying to optimize this Lagrange multiplier because we only need a very large number. We can use an arbitrarily large number for this. However, not all the excitations can be treated as pure, meaning that considering that an excitation is only given by a HOMO-LUMO transition can be sometimes a simplification. There are cases where higher orbitals like LUMO +1 or LUMO +2 or lower orbitals in the occupied space like HOMO -1 and HOMO -2 can be involved in the transition. So we wondered whether we needed to introduce these mixed excitation descriptions in order to have a more accurate description of the overall excitations. So we implemented this option with the idea of impose each of the transitions separately and then combining the kernels obtained for each of the transition. And then we sort of tested it in a number of examples. So I'm going to tell you a little bit about how this is done. So before going into details, what we show was that, for example, for the naphthalene molecule, which is a very simple small molecule, if we consider the HOMO-LUMO transition as being pure, we have an energy that is 4.33. If we consider the HOMO-LUMO transition in naphthalene to be mixed, meaning that we actually, thanks to an analysis based on TD-DFT that I'm going to show you later, and it's also in one of the notebooks, we can see that actually there is HOMO-LUMO transition as well as HOMO -1 LUMO +1 transition and HOMO -2 or LUMO +2 transition with these particular weights. So these weights have been directly extracted by a TD-DFT analysis that has been done beforehand in order to inform this. And what we see, if we consider the excitations to be mixed, is that the energy changes of 0.1 electron volt. So in this particular case, it's not particularly dramatic, but even though this is only 0.93, we already see some effect on the energy. So actually, probably potentially there are cases where considering mixed excitations can be quite important. We applied it to other molecules as well. In fact, actually, our benchmarking set of molecules has been the one of the acins, which are these conjugated molecules. We applied several methods. We investigated the excitations with several standard DFT methods, for example, delta SCF and TD-DFT, and we considered two functionals, meaning that we considered two levels of accuracy, so PBE and PBE0, and we compared these results with our TC-DFT results done with the PBE functionals. As you can see, one of the main, I think the main take-home message from this is that we find that TC-DFT performs in a way that is consistent with the TD-DFT with the hybrid functionals, as well as sort of following the same trend that coupled cluster CCSDT gives. CCSDT is a quantum chemistry method, is considered one of the most accurate methods for quantum chemistry, so it was a good reference. We were very happy with the performance with the acins, and we found that for the acins, TC-DFT performs well both with local and charge transfer states. The reason why we started thinking about constrained DFT was actually because we wanted to model emitters, like for instance, OLED materials. These are materials used to make mobile phones and screens. So we wanted to be able to model this with constrained DFT. We tested constrained DFT on a number of OLED materials. This ACR FLCN is one of this. In this particular plot, we are comparing TC-DFT, which is the central column, with the standard delta SCF and TD-DFT, both with PBE and PBE0, whereas the dotted lines is our reference. And this reference in this case is a very specific, very expensive DFT calculation that we can consider sort of a good benchmark for this example. And actually the very good news from this plot is that, as you can see, where TDDFT fails at describing the delta energy, because as I told you before, TDDFT fails with charge transfer states, TCDFT has a consistent behavior that is comparable to what we get with TDDFT and PBE0. So again, if you look at the MAD here, we can be quite satisfied of the performance comparing TCDFT with PBE0, both delta SCF and TDDFT. So what we showed here is really that TCDFT performs in a sort of consistent way with charge transfer states as well. Okay, so to sum up, this is just a very nice picture that shows that the transition-based constraint DFT can effectively describe excitation energy for both local and charge transfer states. So it can be considered independent from the nature of the excitation, contrarily to standard TDDFT with standard functionals that fails for charge transfer states. So this method has been incorporated in a couple of different workflows because it has the potential to be applied in different situations. So to start with, the most general workflow that we use, and that's actually the one that you're going to see if you want to do the notebook about this particular method, is that we combine information from TDDFT to inform our TCDFT calculation. In which way? So this is a very, very quick sum up of the main steps. We generate the support functions with virtual states because we have empty states now that have to be taken into account. We run TDDFT calculations to get the information about the excitations. Are they pure? Are they mixed? If they're pure, we just run standard pure TCDFT calculations. If they're mixed, then we have to take into account contributions from higher excitation as well. And we can do both. So in this example, I'm summing up what we get from applying this method to another kind of OLED molecule. This is actually a TADF molecule, which is an emitters-based or a slightly different mechanism. It's called 2ZPN. In this case as well, you can see that our method performs quite well comparing to benchmarks, which are these dotted lines, as well as TDDFT with the hybrid functionals. And for this reason, we decided to incorporate TCDFT with fragments approach, like the one that Laura has just presented, to start and do excited states on very large systems, meaning that we can sort of take a morphology for a 2ZPN material, and we can sort of extract big clusters, and thanks to the combination of fragment approach as well with TCDFT, we can try and model excitation energies for emitters in larger systems. And actually, this very idea is the same that we can sort of propose for possible biology applications of the features that we have presented in these last two talks. So Laura has presented a fragment approach. I have presented TCDFT. And our idea was, okay, can these two tools be combined to study possible interesting biological applications? And one of the answers is surely yes, because there's a whole group of applications in biology that are based on emitters molecules, so molecules that emit. And fluorescence and emission is a very powerful tool in biology for things like probing cell tissues or functioning as a flag mechanism for cancer cells and things like that. So everything that is associated with fluorescence can be used as a signal to probe something. So if we can control or predict how the fluorescence and the excitation works, it's a very powerful tool. So one possible application is to study the solvent effects on organic fluorophores to try and control the emission of biomolecules and emitters found in protein, like the green fluorescent protein. Other examples are, like Laura was mentioning, the case of the embedded fragment, where the embedded fragment approach can be used as a guess and in order to reduce the computational cost of calculation of very large systems like proteins or small amino acids. We can continue with the
thing for us to prepare this material, which we believe we want to reuse in the future for many years from now, because this material has been created after many years of work by all of us. There has been lots of discussions, development sessions, paper writing, and similar things beyond those approaches. This is a non-exhaustive list of the functionalities which are available in the different flavors of BigDFT code: cubic scaling, linear scaling, fragments. Beyond each of these functionalities, there is a number of discussions and work beyond, which we will be very happy to share with the community, thanks to the new tools like the Jupyter notebooks, the Google Colab, HPC approaches, and all those things we have briefly mentioned today. To conclude, we have at hand a formalism which is based on wavelets. There are wavelets beyond that, but you can forget about applied mathematics and wavelets. In any case, this formalism enables us to treat the systems at different levels of complexity. We are going more and more, let's say, larger and larger in number of atoms. The larger we go, the less complex we try to keep the description, because the number of parameters will become exponentially complex. So we have this hierarchy of approaches, starting from the cubic scaling BigDFT and climbing up to the fragment approach. We are also working into a QM approach, where different levels of theory can be combined together thanks to those tools. And this hierarchy, when there are overlaps between the regions, can also be brought down. We have seen the examples of linear scaling calculation brought down into cubic scaling calculation. This is very important because as you want to employ an instrument, you would like to know the impact that the approximations you are putting into the system is actually having. With this approach, you can always come back when the number of atoms of the systems make this possible. You can still come back to the unbiased cubic scaling description and compare the different impacts. So this is essentially what we have been working on in various ways in the recent years. And the idea is always to provide indicators, to give somehow a way to the expert user or to developer the way of estimating errors. We have seen this purity indicator, but we also see the WABA fragment indicator, the on-site overlap matrix, the purity indicator of a transition for CCPBioSim. All those things is the rationale which we try to keep along all the way through while doing the new kind of developments. With this idea, one can even move towards a description where you just split your systems into different regions with corresponding interface and calculate different DFT descriptions in the various regions. For some situations, this might reveal helpful. For some, it might not be necessary. The advantage is that we have at hand a tool that would enable us to assess this very question, whether it is necessary or not to increase the accuracy, potentially increase the accuracy of the description. The example of embedded fragments we have been seeing before is an example. One would like to treat a defect in a semiconductor or a defect in graphene. You may want to optimize or not the degrees of freedom near to the defect. And one can go in the direction of a database of degrees of freedom. Laura has explained the cost for calculating a template you don't need to recalculate the same template every time. We may have a situation where the degrees of freedom are stored elsewhere. Wavelets are very good for data compression. Those template-based functions are not that heavy. They cost a few kilobytes, at most 1 megabyte per support function. So those are optimal to be stored somewhere in the cloud and used at will. Those things are things we are kind of trying to explore in the forthcoming years as we sense the opportunities that such kind of approach can leverage. We have been trying to put ourselves in the context of modern DFT codes. No, these ideas are not only let's say, particular of the BigDFT group. They are also similar ideas in the context of fragment molecular orbitals calculation, for instance. And there is a couple of review papers which we have been writing in the recent years. One, most recent, is this one. The previous one is has been briefly presented in the beginning this morning, which is mentioned here. You can always have the slides once again in the BigDFT School project, so I let those information for your convenience to you. The advantage is exactly that DFT can be used to understand a little more about the system. What I would like to briefly share with you for concluding this day is to give you an idea of the potential opportunities you can create in interdisciplinarity in this approach. When one has the possibility of cutting a large system into fragments and understanding the interaction between the system, one has the possibility to discuss that with people that perform, for instance, antibody optimization or bioinformatics approach towards the synthesis of new molecules. This is a toy example I usually show. This is a one monoclonal antibody with its own paratope and epitope region. Don't ask me which is which. I don't know. Well, I know there is one paratope and one epitope. One is X and one is Y of this interaction matrix. This interaction matrix contains the description of the fragments of the two regions, and the color code is telling you which are the fragments that are most strongly interacting. So if you want to optimize an antibody, you may be very interested in knowing which are the regions that interact the most between the antibody and the antigen. And this is similar to what William has shown in the complexity reduction approach, where the different highly interacting, the different hotspot regions of the spike ACE2 complex has been identified via DFT. So you have 22,000 atom systems, you have 1 hour and 12 minutes of your time on 32 nodes of a machine. So this is something you could afford in terms of wall time, and you get information you can play with in order to have a better understanding of a particular structural arrangement of a system. So we were at that time when pandemic came, and I believe we gave you enough element to believe me when I tell you that for us the pandemic was an opportunity, because we were in a situation where the world was kind of listening to new solutions. So we had the possibility to interact with the people coming from communities which are totally different from DFT. And that is also one of the reasons we are here today in a network into which we would never have been necessarily considered in the past years. The reason is that we have the possibility of thinking about large systems and quantum mechanics somehow differently. That enables us a number of collaborations with which we have made a few papers in the last years. One of those is a paper which I would like to highlight, associated to the calculations of interaction of the natural substrates of SARS-CoV-2 main protease with different computational techniques altogether. Molecular dynamics, docking, DFT, as well as experimental techniques has been all merged together in order to have insights into systems like these ones. So the idea is to somehow provide a paradigm of analysis which were not available since now in the community. So essentially, we would like to match experimental data that are associated to communities like bioinformatics, structural biology, experimental crystallographies, cryothem, all those things, with purely computational approaches that were not necessarily at the heart of those methods nowadays. Nowadays, we have the possibilities, thanks to the advent of new supercomputers, thanks to the advent of those new formalisms. So these are, in our opinion, interesting point to address. The reason is that one wants to understand a little more to then re-engineer the system. This is, in general, an inverse problem. You would like to have better drug design, better antibody design, understand how active region of an enzyme may work. So that are questions for which we would not necessarily provide an answer, but at least we would complement the scenario and the panorama with other information that come from totally different grounds. And therefore, they can help in somehow clarifying some points. So our ideas is to somehow go towards a platform where crystal structures, for instance, can be characterized with this method, a platform that can also be used by non-specialists of DFT. We are trying to have a simplified approach for creating input files decorated with those indicators that can somehow be employed to validate the measurement that can somehow bring DFT to other communities that were not necessarily acquainted with the formalism before. So there is an ongoing collaboration with the Newton source in Grenoble that deals with biological systems, which are people that have a little experience in DFT for very large systems. That could be a nice playground for building this approach, which we could call the quantum as a service, but it's not a service in the commercial sense. It's really a service in the scientific sense. So I'm coming to the end of this day. The idea is really to have another way of thinking of DFT, not necessarily just as a force field. It is still possible and suitable to increase the accuracy of DFT methods to still explore these things like the potential energy surface, reactivity reaction coordinates, and similar things which are very interesting in quantum chemistry. But there are also a number of insights that can pop up just from simple single point calculations or a population of them, coming, for instance, from downsampled molecular dynamics trajectories. One caveat I would like to tell at the end is that when it comes to interdisciplinarity, you are really going to discuss with people that don't know everything of what you're going to say. So essentially, my personal experience, for instance, is when I discuss with biologists, is that what it is much more important is the result of your investigation rather than the procedure, which is a kind of the other way around for physicists. results doesn't matter as much as the procedure. That is an example of how a dictionary of languages should be adapted when we discuss with different people. And of course, it is also a matter, as they don't master everything you are going to discuss with them, this is also the same for you. You have to trust them. So it's really important to identify the right guys when it comes to the interdisciplinarity, because in my understanding, you need to have all these four situations. Both of the group have to have their own rigor in their own discipline. Both of the group have to trust each other with respect to this rigor, but you also have to share the same vision about the outcomes of your results. And this vision doesn't come for free, so you really have to struggle. So it's really important to keep committed on your investigation, because when it comes to understand how each other works and think, you really have to spend energies in doing that. So that is to say that going to interdisciplinarity is not necessarily going to simplify your daily life, scientifically speaking, but you can really have so much fun in doing that if you find the right guys. And with that, I think I can conclude. I would like to really thank all the DFT group that contributed to this. We have met in person, for those who are here, Laura and Martina. We met William from Kobe. We met Samuel from Grenoble. But there are also, and we met Giuseppe from Catania. There are also other people that work on those things in different places in the world. So we are a small group, delocalized in various institutions, but